good evening to everybody. Thank you very much for joining today for this webinar. And I'm Priya Werahera. I'm talking to you from beautiful Denver, Colorado. It's called Mile High City. It is exactly 5,280 feet above sea level. It's in the middle of the country. And the beautiful Rocky Mountain Range runs through here. And right now it is end of winter and beginning of spring, so we are still have cold temperatures and snow covered areas. We have 14,000 feet high peaks here, the only state in the whole country which have that many peaks, roughly about 4,200 meters above sea level. And the today's talk is, I have divided it into two parts. Since it is a biomedical topic, I had to cover the disease and the biology and the medicine side first, because otherwise the second half, which is engineering, will not make much sense to you. So, prostate cancer. Okay, let's see how Sri Lanka is doing. That's from the WHO statistics, World Health Organization, from 2022. These are the numbers available. Number of new cases, 33,000. Number of deaths from all cancers, about 19,000. And how do you distribute this during, to various categories? If you look at the first, the chart, the 5 leading causes of cancer, their number, prostate cancer rank number 5. And this is not a disease that the females has to worry about, and this is exclusively a men's disease. And so these distributions show that cancer is a huge problem, and I have worked in this area for quite some time now, over 30 years. And so I'll share you some of the experiences I have here. What is the prostate gland? And most of the engineers probably doesn't know. And this is a walnut-sized gland, stuck in between the bladder and the penis. And the urine passes through the urethra and from the bladder to the penis. And then it has dual functions. One is, of course, passing urine, and then during intercourse, it secretes some fluids that will provide some protection for the sperm. And I usually call it the gatekeeper gland. Anyway, so the cancer statistics in the United States for this year, the 2026 estimates are in the figure here, and you can see we are going to discover about 333,000 new cases of prostate cancer. That's about 31% of all cancers, and it is also the second leading cause of death. The number one, of course, is the lung cancer. And so we are offering about 1.3 million or even little more than that screening tests for men in the country. And this is going to cost us big time. The screening is $3 to $4 billion, and it will be actually more by now. And the treatment is, even more than that. One of the issues is, if you detect the cancer early, it has a very high cure rate. So about 80% of the men who have prostate cancer is not going to die from the disease. So only about 4% that's going to die. So most of the men get overtreated and leaving for poor quality of life. Actually, they can be left alone. So what is the strategy if you are a man? Early screening. It's always save lives. And if you have a family history of any cancer, just not prostate cancer, mother, father, grandparents, any uncles, any kind of cancer, especially if it is prostate cancer, then you need to be screening for this. If you are 50 years and old, it's a simple blood test called PSA test, prostate specific antigen test. If it is more than 4 nanograms per milliliter, the levels, then you are advised to get a biopsy. And of course, you can do a digital rectal exam. Biopsies are the gold standard for this cancer, and the clinical detection rate is something about 25 to 35%. The irony is almost more than 50% of the cancers are missed during the initial biopsy. So it's a challenge, so how do you pick up all these men on the first time? So the biopsies here is a transrectal ultrasound guided biopsy. the patient is placed in the, on the sideways, and then you put the transrectal probe, ultrasound probe into the rectum, and then you can actually insert the needle through. There's a little guide inside the in the probe that you can actually do biopsies. And gold standard, still the gold standard, you take biopsy course, bring the tissue out. You take about 10 to 12 biopsies, and then the pathologists review these cases and then tell you whether it is cancer or not. On your right hand side, you can see a slide. There are two glands, one gland on the top, one gland on the bottom. One is cancer and one is not. Can you guess what it is? So usually the normal glands have several types of cells, basal cell layer, then the luminal epithelial cell layer. And, in the top gland, you have the basal cell layer, but in the bottom gland, you don't have it. Those black lines, we have a special stain to show that the basal cell layer present in the gland on the top. When the basal cell layer is gone, that's called cancer, in a very simple term. So just so that you know, that's a cancerous gland. So biopsies are taken randomly. When the patient comes in, the doctor doesn't know where the tumors are. You just keep sticking needles. And if you look at the picture on the right hand side, you can see the biopsies taken, and then this is a simulation I have done, and nothing touches the tumor in the middle in green. So the biopsy cores are taken, but this tumor is now missed. So it is subjected to sampling errors. So the main issue is more than 90% of the cores are benign and only, very few cores comes positive for cancer. And as I mentioned before, more than 50% of the important cancer, ones that we want to treat, are missed during the initial biopsies. So the clinical challenges are to increase the diagnostic yield. You want to get a higher yield from your biopsies because you're only going to take 10 to 12. If you have, only one core positive out of the 10, that's not a good yield. So the second one is, of course, we want to find those cancers that we really need to treat. I mean, those guys are going to die if you don't do anything. So optical spectroscopy. So now we're moving into the engineering side. So this is the area common to both. It has been around for a long time. The light photons interact with biological tissue in different ways. Tissue absorbs, tissue fluoresces, there's scattering, variety of methods. And optical properties are determined by the molecular composition and the cellular morphology. And if there is a malignant growth, there are changes that are happening in both the molecular and the cellular levels. So we can use these changes in the biology of the tissue to diagnose diseases and accurate detection. And you can use fluorescence, absorption, scattering, Raman scattering. There are a number of methods available for this. And when I first started, there was nothing available for prostate cancer because of the heterogeneity of the disease. And then, of course, the accessing this gland is not trivial, so you have to, the only one path available is through the rectum. So it's not readily available, techniques to go and do this. So you are looking at the electromagnetic spectrum and the region of interest. So one of the problems we have is the two large component in your body is blood and water. Both absorbs photons. If you put light into them, they absorb. So the yield, if you pump 100 photons into the tissue, only about 10% of those actually go on to scatter, fluoresce, and all those things, the rest is all, absorbed by these things. However, there's a small region called the therapeutic window between 600 to 1000 nanometers. It's largely dominated by scattering, and you can see the curve here, and the absorption due to oxydeoxyhemoglobin is very minimum, and then so is water, and then water is going to pick up from there onwards. So if you try to work some of your optical spectroscopy stuff in that window, you can get a slightly better yield, okay, quantum yield. out of the photons. Now, unfortunately for fluorescence, the fluorescence is taking place in 300 to 500 nanometer range. So if you want to do fluorescence, that's where you have to go. All right, fluorescence spectra. So most of you know there's a ground state, and then the photons, the electrons absorb some of the energy from the photons. It go to an excited state, and then they release the energy at a different wavelength, which we call fluorescence. Some you can see into the naked eye, some you cannot. But actually, if you have detectors that can pick up this signal, then you have a fluorescent signal, although you cannot visually see this thing. Now, there are various, acids, structural proteins, enzymes that fluoresce. That means absorb photons and then emit another signal. The endogenous fluorophores, some of them are NADH, FAD, tryptophan, collagen. If you look at the table, there's a whole bunch of those. And in order to get a fluorescent signal, the excitation, the photons have to be in the 250 to 450 nanometer range. So you pump photons in that range and then you get a fluorescent signal. Very weak signal, by the way. And of course, if there is a malignancy, the concentration of the fluorophores change. How? What happens? For example, if you take collagen, it's a structural protein. If the tumor begin to eat into it, and then the collagen levels in the extracellular matrix drops down, reduce. So compared to the normal cells, normal glands, there's a less collagen in cancer cells. Same thing with tryptophan. It's an amino acid. It's called tryptophan catabolism. The levels go down. NADH, on the other hand, is different. It's an enzyme, and the cells process glucose, and cancer cells divide very fast, and it needs more energy. So consequently, NADH levels goes up when there is a malignancy. So we have measured this thing, and you can see in the graph, and we have we have taken excised prostates, and we have analyzed. You can see very clearly the tryptophan signal, collagen and NADH corresponding signals. And there are various other ones also, but these three are the big ones for prostate, at least. And some of the other sites like breast, lung, liver, they might have some other fluorophores that might be more useful for diagnosis. Here is a classification table. If you use, I have done some principal component. I will get to the engineering part a little later. Just for the sensitivity, specificity, negative predictive value, if you use the tryptophan at 280, 290, 300 nanometer excitation, collagen, and also NADH, the sensitivity is range from something like 70 to almost 86, almost close to 90%. The specificity is also very good. The negative predictive value is very high. You can see. So this is, of course, outside the body. So the prostate is sitting in my lab, so we are testing it. No blood flow. Now let's look at what happens when the blood comes into the picture. So here is a plot on here that we have taken in the surgery room when the prostate is still intact. So there's a blood flow, and then you bring the same prostate into my lab, and then we take the measurement. So on the left-hand side is when the prostate is inside the patient, and on the right-hand side is the prostate removed, sitting on a table. What you need to see is the difference between the benign and the malignant separation when it is in the lab ex vivo side. You can see very clearly. Now you can classify this, benign and cancer. We look at the separation, tryptophan, collagen, NADH. But look at when it is inside the body due to the absorption of the blood. That separation is very small. So your AI algorithms has to do over time to classify when we have this, when we want to do this in real time. So this is something to keep in mind. That's why I said about the absorption due to blood and water at the very beginning. So this is the impact. So you can see in the lab, very easy. You can do this very easily, but when it is inside the body, it's a whole different ballgame. Scattering. Okay, so you put the photons, the light bounce. It's a very strong signal. It tells the cell nucleus, cell boundaries, glands, it's a very strong signal. And consequently, because it depends on the architecture of the glands, the tissues, the cells, it is sensitive to the cancer grade. So the grade tells you how aggressive the disease is. Higher the grade, the more aggressive the disease. Right now, we use the Gleason grading system. We have gone through several revisions. Right now, we have Gleason 3, 4, and 5. So there are grade groups and everything else, but the patterns 4 and 5 are very bad. If you have Gleason grade 4, 5 disease, the prognosis is very poor. On the other hand, Gleason grade 3, it has some much better prognosis. Anyway, so here is how the scattering spectra looks like. Again, I have basically said benign, and then low grade is Gleason grade 3, and the high grade is 4 and 5. That is the scattering tissue, and there is a mean scattering range here around like 600 nanometers, and you can actually classify very nicely. But again, the accuracy between malignant and benign, that means all cancer versus benign, is pretty low. But look at the high grade and the benign, 76%. So this is what we want. We know identify these high grade cancers. So this will play a role in our project. So with all of that background, now as an engineer, you wanted to put something together, some pieces together, so you can actually do a diagnosis in real time. So from all of that, and keep in mind, in the clinic, you had to do this very quickly. You have very limited time. You can stick a needle and hold on for 30 minutes. Okay, that's not possible. So you have to stick the needle, biopsy needle, into the prostate, and then you have milliseconds. So you want to keep your excitation sources to a manageable number. So what we have done here is, for autofluorescence, we have picked up 280 nanometer, which will excite tryptophan, and we use a single 340 nanometer for both collagen and NADH. The elastic scattering is between 400 to 800 nanometers. So that's sort of like a decision we made, get going, because we wanted to make it real time, because of the clinical, we don't want to disrupt the clinical workflow. So the other things you have to keep in mind, of course, is going to be a commercial product. It has to be able to seamlessly integrate with the existing workflow. It should not make a mass changes into the clinical room to do this. And a positive healthcare outcome for the patients. And it is, can be expanded. This is ideally for big data, for future expansions. And then, of course, the government, the healthcare system has to reimburse for these things, for AI applications and all those things. And for the technology should be able to readily extend it to other sites as well, liver, breast, all of those things. So ClariCore. So it has a console and a biopsy needle. That's basically it. And so the optical biopsy needle, it has a fiber optic cable in the middle. And then there's a handpiece which will advance the needle in steps of 1 millimeters, and a console that will take the signal back and tell you whether it is cancer or not. Here is the optical biopsy needle. And the needle consists of two parts, outer needle and the inner needle. The inner needle have a hole in the middle. That's where all the optical fiber comes through. And at the tip, you have the where you can shine light and get the tissue excited. And then it also do the reading, same fiber. So we have a sort of like a switching mechanism at the back end, single fiber, 200 micron single fiber, with a 20 nanometer aperture. And so you excite the tissue. Let us say you turn on 280 nanometer LED and then read that back. Then you open 340 nanometer LED, read back, and then you have a broadband light source. From 400 to 800, take the signal. All of these things for a single, for these three measurements, all done under about 100 milliseconds. One measurement. So there are 18 measurements we do together. And here is so there's a stepper motor inside the handpiece that will advance the inner needle in steps of 1 millimeter. I'll show you a video once we get there. And once you gone through the length of the prostate, 18 millimeters into the prostate, and then you press the button, and the outer needle will come and cut a tissue piece, the one that you classified, the one that you measured. So the outer needle is the one that the cutting mechanism. If you go back here, you can see, this will cut the piece of core, and then there's a little notch here, the sample notch we call it. So the core will be in there, and then you take that to the pathologist. This is the ClariCore console. Fairly straightforward. There's a motherboard, and there's a patient isolation circuitry because you don't want to electrocute the patient. And then the hand piece and the power distribution board, which is the one that controls the biopsy needle. Then the excitation source, which are the light sources that you send to the fiber, and then the spectrometer is the one that leads the signal back. Here is the console specifications. The motherboard is an Intel Core 5 i5 processor, and the memory units are standard things. And then there's a master controller, and the excitation sources are all LEDs, light emitting diodes, about 10 milliwatts power. And we stayed out of lasers because getting FDA approval is not easy when you have a laser. LEDs, low power, 10 milliwatts. Fortunately, you don't need a lot of power to excite these tissues. It doesn't matter. I mean, the quantum yield is very poor, as I told you before. And LED have enough power to, excite the tissue. And the spectrometers are there. I think we bought something from Avantes. I think they are off-the-shelf products, and we put together the whole thing. And I'm not going to even discuss the software for this thing. That's a whole nother project. You'll be spending the whole day with me if I started on the software, because there's a graphical user interface, and then everything from driving the needle to data collection and variety of other things. We all develop all of those in-house. Okay, actually we got a company to come and work with us, so it's a whole big software project. And here's a block diagram, the same thing. And as you can see, the power distribution boards, and there are fans, and and on this side you have the excitation, and you can use a USB thumb drive to collect your data. If you want, you can do through the network too. And you might ask, how much did it cost, by the way? Yeah, it costs more than a BMW car. Of course, these are R&D, and they are all involved. So yeah, it's about, we created like about a dozen of these units. Hand pieces, we created a lot, actually. Biopsy needle, because they are disposable, by the way. Once you use the needle and the hand piece, that is a throwaway piece. About 60,000, but the target was to bring this, if you go into the commercial basis, between 5,000 to 10,000. This was back in, a few years ago. So the prices may have changed, but once we finish the development, that is how much it cost us. Clinical arrangement. So this is a schematic. Unfortunately, I can't give you any photographs of the the patients in there due to privacy issues, but this shows how the ClariCore, again, you have a transrectal ultrasound probe, and just as the biopsy needle, it goes through the guide into the prostate, and the console is there. Then the doctor press the button, and then the needle begin to advance automatically, because I told you there's a stepper motor inside the handpiece, and then you can collect the data. Let's look at how this is done. This is an animation, but it should give you quite an idea what happens, what goes inside the machine. So it says there's a difference between normal and malignant tissue. And there's the prostate, and then here's your needle. Right now, doctor inserted the one. Once now you press the button, the stepper motor takes over, and then it's going to go in steps of 1 millimeter. At each step, it classifies the tissue, that 1 millimeter segment. So there are 3 light sources shine at that point. And then once it is done, you cut a core and bring it out. Now, we were very fortunate to do a multi-center clinical trial, and all of these centers participated. John Hopkins, of course, you probably would have heard. They were a big part of this study, and the urology center in Colorado, and some couple of private groups. So we were able to get 146 patients. 2 dropped out. For our initial clinical trial, we got the the FDA, the Food and Drug Administration approval for the device, and we did 44% men were initial biopsies, and 63 of them were initial. They were first-time biopsies. About 56% they were repeat biopsy guys, meaning they had biopsies before but didn't detect any cancer, so they were also included in the study. And the numbers are here, and then these are the baseline data. As I said, the age is about 66 median. The PSA, the one that I told you about, the blood test, all of them had the blood test done, and the median, the mean is about 8.3 nanograms per milliliter, and you can see it is elevated above 4. That's why they were subjected to biopsies, and the BMI. And we wanted to make sure. Sometimes when you cut the core, they exactly doesn't come as 18 millimeters. Sometimes they are short pieces due to some mechanical operation. So the tissue sometimes doesn't cooperate very well with the mechanical devices. So you get fragments, tissue fragments, but you still have to classify. So about 33 patients were diagnosed with prostate cancer out of the 47 patients. 16% of the biopsy cores, had cancer, and 85% of the cores have Gleason grade greater than 2, meaning high grade cancer. Okay, in node pattern 4 and 5. So you might ask, okay, how did you guys do this? So on the top here, you see the biopsy core, and each millimeter is classified. Every millimeter. Here in this one, we only had 15 millimeters. We know which end was proximal and which end was distal. When we are taking out, we put a little bit of ink on a one corner so they know the orientation of the biopsy needle. So the pathologist painfully classified every one of the millimeters of this. You can see the first 1 through 9 bins, we call them bins. 1 millimeter bins were benign. There's no cancer. The bin number 10, 11, 12, 13, and 14 have cancer. The 3 plus 4, the pattern 3 and pattern 4, 80%, 100%, 100%, 100%. So we get this information. So we did this for every biopsy core, by the way. Every biopsy core. Otherwise, you can't correlate with the spectrum. So every one of these bins have a corresponding spectra. 3 spectra, actually. So one is the fluorescence spectra of the tryptophan. You can see the red indicates the cancer, the blue indicates the normal tissue. Actually, this is an average I'm showing you. And I haven't shown you the 340 signal here, but I am showing you here the scattering signal. By the way, the shape of this is the LEDs we use. And again, you can see the blue one. There's some separation between the blue and the red, which is the benign and the cancer tissue. So in the algorithm, we correlated every bin with the corresponding spectra. So we know when we have the data comes in, which bin is cancer, which bin is benign. And then we have 3 spectras. And so the tissue classification algorithm. So these are the things that you probably very much interested. So I'm giving this for the fluorescence spectra, but this is the same thing we do for all other things. We take the spectra, fluorescence spectra in this case, and then we take the histopathology, which is the classification, benign or cancer. If it is the cancer, we know the grade and all those things. So we had to do a background subtraction. Remember, I told you when the needle is inside the body, that separation between the benign and the malignant is very small. So if the noise flow is very high, that signal begin to get even smaller. So you do a background subtraction, then we decide on a signal to noise level. And if the signal to noise level of that particular signal for somehow doesn't meet the requirements, we just take it out because it is already corrupted. Then we do a data smoothing interpolation. Remember, the spectrometer doesn't give you a smooth curve like this one, like this one. This is actually I have interpolated. It's the output of the spectrum. The spectrometer is kind of like very wavy. So once you do the interpolation and some smoothing, you get a very nice curve. But so we had to do that. And then, of course, normalization. That is an important aspect. Now, you might think the fluorescence level of individual patients are same. They're not. There are levels that are different from patient to patient. So they are called, interpatient variation. Second one is, I told you the disease was heterogeneous. So same tumor, same cancer within one patient have different behavior. So whatever the fluorescence level you are measuring on one location may not be the same on the other location. So there is inter and intra-patient variation. So how do you get around with this problem? Because you can't have an absolute level. Okay, if the fluorescence level is so many AU units, it is cancer. If it is below, it is cancer. Above, it is normal. You can't set up a level like that. So it's a relative thing. So what you do is you normalize. What I did here was I normalized to the area under the curve. So each curve is normalized to itself. So meaning the area under the curve is 1. All of the spectrums, the fluorescence spectrums, the scattering spectrum, they are all normalized to 1. There are other ways of normalizing too. Now, I believe you have done principal component analysis. It's a data reduction scheme. So partial least square is also a special way of doing the principal component analysis. Usually when you do the principal component analysis, if you have an X and a Y, the straight PCA basically optimizes the X. It doesn't look at the Y. It tries to come out with the components, principal components 1, 2, 3 on X, but not the Y, the output. The partial least square analysis, it is looking at both X and Y to pick up the principal components. So you run through that, you pick up your principal, because you don't want to feed this whole waveform into your AI algorithm. So you pick up what are the key principal components and then use those. In the algorithm, I gave as an example here as a support vector machine, but you can put anything, any AI thing, random forest, whatever you want to put it in there, you can put it in the algorithm. And then validation, you can do a leave one out validate, then an external validation. You separate. There are a number of ways you can do the cross validations to see how good your data is. And so we have a cutoff of signal to noise ratio of 6. We discarded. We ran partial least square analysis, and we use support vector machines in most of these cases, but you'll see I have done some other work too. So training and validation. So you take about 80 patients and did the training and then testing with 62. Okay, so we separate, when we were doing this whole clinical trial, we agreed, okay, we're going to take the first 80 for our training purposes, and then the other 62, we are going to keep it separate for testing. So we use several classification methods, support vector machines, random forest, booster tree, gradient booster tree. Logistic regression is, I thrown it here because that is, just to see what the standard logistic regression do for classification. So it does about sensitivity of 85%, but you can see, now remember, these are all training and validation, same data. Okay, we have not done the testing yet. And negative predictive value is very high. We know the NPV is a very important thing, because if NPV is higher than 90%, if your signal says green, meaning it's normal, you can pretty much be assured that it is normal. Now, pretty good results within the training data set. Now, testing. performance on testing data. So you can see that the sensitivity slightly drops, but it is still at very high 80s sensitivity and specificity, because now this trainings the testing data set is not part of the training data set. It has never seen it. Is like the whole new patients are coming in, and now you are asking your algorithm to do the classification, and it's still very good. So we have significant challenges facing this. Any questions up to this point? You can. I will get to all of these at the end of the presentation. So we have done this. It took us, of course, there's a lot of money involved here, but getting a device into the healthcare is, you have the FDA approval and nuclear clinical validation. That takes a chunk of time. I mean, in addition to the R&D part, this whole clinical validation is a whole big ball game. You need a lot of money. Then, of course, you have to make sure market penetration, how the doctors adapt to these new devices, short-term, long-term goals. Then you have to train the doctors, the staff with the new device. And of course, the economics of doing all those things. You need to get some money back. The doctors need the money, and then the devices, as a company, we need money. And then, of course, we had to develop this thing I told you about, the big data. So once it is in the clinical use, you can begin to collect data and then begin to refine or revise your AI algorithms. So in summary, you can use the optical biopsy needle for diagnosis. We know that, it's already taken care of. The doctors like it, and you can do this in real time. When you put the needle moves in, tick, tick, tick. You can see the classification, and it reduces sampling errors. Now, the doctors have the luxury. If you don't get a cancer, you don't have to cut the core. You can just pull the needle out without cutting a core, and you can go to another place. And if you have an MRI image that shows there are some areas, the MRI says, okay, there is cancer in the prostate, you can go to those areas. And the most important thing is, of course, you might ask FDA, what are their number one concerns? Safety. Number two, safety. Number three, safety. That's all they are concerned about. So there were no serious adverse events, and the pain score, which was very tolerable out of a 10, it's about 2, which is standard for biopsies. And this also will reduce the cost. Right now, the government, the US government, the Medicare have said you're going to get paid $700 for biopsies. You can take 10, you can take 50. 700 is the number you're going to get reimbursed. So you want to make sure that 10 to 12 cores give you the biggest bang for the buck. So you don't want to take 50 cores and then eat the cost. You want to make sure that 10 to 12 cores can diagnose the disease. So I have a lot of people to thank. This is not a one-man effort. And as you can see, a lot of doctors participated in this one. And this one I have highlighted here, Dr. Patin. He passed away in 2023. He's one of the one of the key figures in the urology area in the country. And then this Amir Tehrani was the CEO of the company we call Precision Biopsy, and his team worked their butts off to get all of these things done and have this thing worked out. Of course, we hit some barriers. Number one is the COVID. Number two are all kind of wars. Ukraine war. Now we have a Gulf war going on, so the funding becomes very critical. But as you can see, there's a big team here for me to thank. And I have given you some references. I'm the principal investigator for this whole study. I'm very proud to say that as an engineer who started at University of Moratuwa, electrical power engineer, end up being a biomedical engineer. I hated medicine and biology. But the moral of the story is don't hate things too much, then you will end up doing exactly that thing you hated. But the funding is on the right-hand side, as you can see. There's about, right here, about close to 40 million, but that is the direct funding we got, and then this Sam Bigam Foundation that are indirect funding. So probably the total project cost us about close to more than $50 million to develop this whole thing. And I think I'm doing very well with this timing. I can show you another slide. I'll come back to it. I put up these slides so that I can show you. You might ask, why is this so important? I told you at the beginning, not every cancer is going to need to be treated. More than 80% of the cancers can be left alone, or we can say minimally treatment. So right now the treatment is you take the whole prostate out. You might ask, isn't there a risk? Yes, the quality of life. Otherwise, people can live. A man can live without the prostate, but it impacts the quality of life. You have to have a wear a diaper and urinary considerations and variety of things. So, if instead of taking the prostate out, supposing we identify where the lesions are, we can treat the lesion, not take out the prostate, just go and ablate the tumor, which is done, in other sites, but not for the prostate. So we said, okay, in theory, how do we going to do this? Okay, so if you look at an MRI, there's a lesion there, then you go and, try to sort of localize this using our device. So here is another video how we want to do this. Now, we have done this at a couple of clinics. So we call this 3D mapping. So the patient is there, we look at the MRI, and then we have some idea where the lesion is. And now there's the MRI, and we have, the lesion on the patient's right-hand side. So we do a planning. We're going to take some course from around this area. This is the planning. And then you use the ClariCore. And you do this to a template. And instead of transrectally, we do transperineal now through the perineum. So you keep taking biopsies, and then you are trying to do a 3D map of where the lesion is. And you take the course, and you can see now the ClariCore confirms the presence, the cancer, and we know exactly what coordinates. You can see the coordinates. Now you can put cryoprobes or electrodes or anything into the prostate and ablate right there. That's where the lesion is. You can ablate it. And so we have done this at 2 centers, only about 14 patients, and we have very successful results. So that's how much far we have gone. And we have another company now that I'm working with now. Hopefully we will find some money to get this thing to the next level. But that's where we should be. And thank you very much for listening. And I believe we have plenty of time for Q&A. I'm pretty sure I can go back to, if you keep track of the slide numbers, I can go back to those slides and answer any questions you have.
Yeah, so thank you very much, Dr. Werahera, for that highly insightful and informative presentation. So now let me introduce our speaker of the day. So we are really honored to have Dr. Priya N. Werahera with us today. So Dr. Werahera is a research associate professor of pathology and bioengineering, retired at the University of Colorado Anschutz Medical Campus in Aurora, Colorado, USA, with more than 30 years of experience in biomedical imaging, optical spectroscopy, and bioinstrumentation. He has made significant contributions to cancer diagnostics and medical imaging technologies. His research area has focused extensively on prostate cancer diagnosis, including the development of specimens and advanced computer algorithms to create 3D models of human prostate, the study of tumor growth rates, and biopsy mapping technologies. Dr. Werahera has also been actively involved in developing innovative optical biopsy technologies, including minimally invasive systems capable of detecting prostate cancer in real time. Currently, he also serves as the Chief Science Officer and consultant at Preview Medical INC. He is also a senior life member at IEEE. So once again, thank you, Dr. Werahera, for joining with us. So now we can move on to the Q&A session. So if y'all have any questions, y'all can drop them in our Zoom chat.
I can see there are two participants raise hands. What do
Yes.
I click, or who's gonna?
Uh, Participant can unmute themselves and speak if you want.
Hi, Dr. Priya. Thank you very much for the presentation. And it's a novel region, I think, on the engineering perspective. So my question is more on the engineering side. So when designing this kind of medical device, what kind of certification and standards which need to follow on the design stage, specifically for this kind of medical devices in USA? Thank you.
Yeah. So again, as I said, it's the safety. It's a safety issue. I mean, the efficacy comes later. Whether the device is effective or not. That's not the issue. First thing is you want to make sure do no harm. That's the important thing. So from this kind of a device, the harm can come from the light sources you use. If you use a laser, now that's why we stayed out of it. Of course, when we were doing this, somebody said, if you say the word laser, there is a Christmas tree at the FDA head office that light up, boom, with a red light, flashing light, lasers. Because the lasers are, a big risk, if you don't do it properly. So because of that, we went ahead and chose LEDs, 10 milliwatts, so we could get around one. Second is the spectrum we are capturing these signals, and you saw that it was 280, 290 nanometer excitation. That is close to the so the UV band, ultraviolet band in the spectrum is split into like 3 UV A, B, and C. That 280 nanometer excitation, is known to cause cancer. So if you do some literature search, that particular wavelength is a cancer causing wavelength. So we had to go through some hoops with the FDA to assure that at this rate, we are putting on like 10 milliwatt LED, and the tissue gets, after going through the circuitry, through the needle, all the way to the tissue, is roughly like 4 to 5 milliwatts. It's not gonna cause cancer. So if you stay in sunlight, they say don't expose yourself to direct sunlight and it can cause you skin cancer. That's that wavelength, the primary wavelength. So that's that's the second one. So those are the two light. The third one is, of course, it has a stepper motor in the handpiece. Now, the standard needle, the doctor controls it. Okay, the doctor puts in, so it's all manually done. So there's no safety issue. But now you are handing this over to an electrical motor, then there is a risk of getting electrocuted if a bad signal comes through, because it's metal. And then if the stepper motor goes crazy and then can go through and without stopping at the after 18 millimeters advancement, it might puncture the bladder. So there were a number of uh, safety issues that we had to show hardware wise and also software. It's a big process, but those are the key ones that we had to meet. And then after the safety concerns were taken off, then the performance standards, the FDA gave us a performance one. 90% NPV and 50% positive predictive value. Now you might ask why they gave those two numbers. The NPV and PPV are part of the disease prevalence. That means in a general population, how many of these guys have cancer, and then how is your device going to perform in them? So those are the numbers. They were not really worried about sensitivity and specificity. So those two were the performance numbers. So we meet 90% NPV, negative predictive value, and 50% positive predictive value, which we met, by the way. And so those were the bigger concerns. Again, as I said, safety. Those are number one. So I told you the safety issues we had with the light sources, the needles, and you can see there are in the schematic, there are isolation boards put on into the console to make sure there's nothing electrical, no shocks, even a small DC signal getting into the patient through the wires, through the cables. So those were the major concerns. And then the performance.
Yeah. So thank you, doctor, for the answer. And in the chat box, we have another question. Thank you very much for the very informative presentation. And is the biopsy taken to validation of the optical results?
So in order to develop an AI algorithm, you have to have training data. That means you need to know the answer. So you have taken a biopsy, and you need to know the answer in order to tell the algorithm, whether it's a support vector machine or a neural network, whatever you're going to use. So in the training data, so we took the, if I can. So the biopsy, so when we take this biopsy, of course, I said it goes to 18 millimeters. Now, there's only 15 millimeter length here. So we had to discard the other 3 because we haven't collected, our device didn't cut that piece of tissue. So 3 millimeters, this is 3 millimeters short. So that spectrum and everything is thrown away. So in this one, here is the biopsy. And this is an H&E slide, by the way, hemotoxin eosin slide. So the pathologist will, so we did this digitally by, we scan all the slides, and then the digitally pathologists classified all these things. So so each bin is a 1 millimeter in length, and call them benign, there's no cancer. And then of course, if it's cancer, they gave the percentage. How much of a percent in that bin is cancer? 10%, is 80%, 100%, and 20%. And then classify that. Now, why is this important? Because the optical depth of penetration is going to vary. It could be from few microns to about centimeters when you come to that therapeutic window. So this is almost a highly localized measurement, because in the region we measured this signal, the photons doesn't penetrate very far. We were looking at about a couple of hundred microns. So this measurement is very accurate. And then, yes, as I said, then each spectra, you can imagine, they came in YAML files. The data files are fairly big for each spectrum. So we had to correlate this, and then we have created a separate database to do this. And yeah, every bin have a spectrum. 3 spectra, actually 4. There's a dark current, which we call the background. Before everything, you turn off all the lights and take a measurement. And then, of course, every one of them have the 280 excitation LED, 340 LED. So 280 have the following tryptophan, 340 follows collagen and NADH, tracking those, and then the broadband light source. So we had 5 spectra to work with for each millimeter. So we had we had a lot of data to work with, and then that made a big difference.
Hi, Dr. Werahera. I'm calling from Toronto, Canada. Name is Sivanalaya. And first of all, let me congratulate you as an alumni of Baroto University myself on your achievement. And my quick question is whether your piece of equipment can be adapted for other forms of cancers too, to test for other forms of cancer.
Yes, so it's a modular design. So now remember, this particular needle will not be able to do, like, for example, breast biopsy. Okay, the breast biopsy needle is slightly different. So, and then if you want to do a liver biopsy, it's a different thing. So, however, the console, the excitation module is designed, it's a modular design. So first thing you need to do is collect data and figure out what are the important fluorophores. I mean, the tryptophan, collagen, they are always there, but some other sites like liver and lung, they might have other fluorophores that are different from benign to malignant. So you identify that light source, and then you can put it in there. And so it's a matter of swapping the LEDs on this end. And then the fiber path is fairly straightforward. And then, you can make it a probe. I mean, you don't have to take a biopsy. You can take it out to a probe, and then you can put the probe into a location and see whether there is cancer. You can do this for kidney. So the needle has to be the change part, but then the algorithm, of course, the AI has to be developed. Okay, I mean, you need to collect data from whatever that particular site. So whatever you develop for prostate will not work for um, kidney and lung and breast and all those things. So you have to go and collect the data. Now, the data collection is not trivial. And, in our case, of course, we are very fortunate. The prostate gland is very small, and you can bring it to my lab and put it on a bench and do the testing. Now, if you try to do this with liver is a very large organ. and then, of course, there are risks also. You have to screen these things before you bring them into the lab, because you can have hepatitis C and HIV, and there are a variety of other diseases. So when you're working with human tissue, you have to be very careful. But for granted that each individual organ have to have its own little data collection first to identify what are the fluorophores. I think these three, the tryptophan, collagen, and these things will work. Now, there is another spectroscopy technique called fluorescence lifetime measurements. So that is how fast the fluorescence signal decay. So you can use a titanium-based light source and then put that and then excite the tissue. Let us say for the prostate, and you study how fast the tissue, the fluorescence signal decay, called the tau, and tau is shorter for cancer and longer for benign. So that's another method. That's a different method. So yes, so we develop this thing in a modular way so that you can plug in, switch light sources and add more capability. Now, Raman scattering, that's a different beast. And Raman spectroscopy is it's very, very promising. So the Raman is a very, very weak signal. And you are looking at something called Raman shift. And I don't want to get into more details here, but we have investigated some of those things too, but we have not yet adopted into the clinical setting yet because of the the very weak, it needs, the spectrometer we used in this one will not work for Raman. I'll tell you upfront. And Raman spectroscopy is very expensive. Even a laboratory unit is cost $20, $30, $40,000. So yeah, so we have developed this keeping in mind so that we can extend it to other sites.
Thank you. Thank you. The name you mentioned, Raman, is it named after Sir C.V. Raman?
Yeah, the Indian the scientist, yes.
Oh, okay. Yeah, interesting. Very good. Thank you. I think you should give some tips to the young engineers who are watching this program to
Correct. I
know.
mean, when I started this, I'm a computer network guy. I mean, I did my PhD in computer networks. I mean, I'm the one who
Oh,
first
okay.
modeled the fiber distributed data interface, FDDI. If you look at my papers, my
Okay.
thesis, everything is in networks. And then, of course, I was working at CU, and these guys in the medical school got me excited about this one. They gave me this as a challenge. I mean, they challenged me. I mean, I I never go away from a challenge. They basically say, well, if you are such a good
Right.
engineer, how about
Interesting.
modeling the prostate in the computer? We're talking IBM PCs those days, 250 megabyte hard drives. I was like, no
Right.
kidding. Yeah, I modeled
Right.
the prostate in the computer digitally. It took me 2.5 months, and they
Interesting.
hired me. Yes,
Interesting.
they said, okay, we want you. And I said, what is an engineer going to do in the medical campus? And he goes, oh, come on over. We know how to keep you busy. And
Interesting. Interesting. Was Professor Idrua one of your lecturers?
yes.
He was my
He
batchmate.
did
He
power
was my
systems for
parallel
us.
batch.
Yes,
He was my
yes,
parallel batch. I did civil
ah,
engineering.
yes. He actually finished his doctorate in
Cambridge.
UK,
Cambridge.
and then he came
No, he
and
Imperial College.
third year
Yeah,
did power systems too.
he went to Imperial College for his PhD.
Yes.
I was at Imperial too during that time. Thank you for your talk. Very good.
Thank you very much. Any other questions?
Another small question. Actually, you are currently in the R&D phase. So when you are developing it to the commercial product, what are your plans to reduce the cost and reduce the production time and those things?
Yes,
And also, I think
they call it the scaling. I mean, there are industrial terms for this. You hand over this whole thing to a company, they know how to scale this thing down. So right now, a standard biopsy needle here costs about $50. The needle, the manual needle we use is about $50. So you don't want your biopsy needle to be $1,500 because you're going to throw it away. so this needle, the biopsy needle, this one. So we have plans to manufacture this for about between $100 to $200, and it's not, so that's one target. Now I said this unit, this one cost a BMW. The company target is to make it, of course, $5,000, which is kind of pushing it a little harder. Anywhere between $5,000 to $10,000 is good, and the model is like the razor blade thing. You know what I mean? You give the razor free, and then you charge the blades. So we were going to give this console free to the clinic, and then charge for the needles. Or give the console at a discounted price, because the needles are the one that makes the money. Everyone $100, $150, $200 range. So that will be the profit margin. So there are various models, but those are the target numbers. So the standard current practice, $50 for the biopsy needle, and our needle will be between $100 to $200, because it has, and we have the reimbursement from the government for this product. So we got the reimbursement codes and everything else. Now, of course, every time I sit down and try to get this thing working, if there's always a war breaking up, if there's not an epidemic, some kind of a disease, and so getting funding it's always a challenge.
Yes, sir. Thank you, sir, for the explanation. Actually, you have some servo motor with that to go for 1 millimeter intervals, increments. So with the difference in the tissue hardness, how do you like ensure the precision of the movement?
Yes, so this is another very important question. How do you correlate this? Because the engineering side is very precise. I mean, but the biological tissue, I mean, all bets are off when you do this. So one of the things we always sort of argued, and this has always comes up as one of the issues. Now, if the core is benign completely, that's no issue. So the normal signals are fine. It is the cores that have cancer that always gave us some headaches. So one of the things we did in the very first cut was we narrowed down the data for those that we know for sure are cancer. And although you start with 80 patients in your training data set, that data set was reduced to something that was, that we means the pathologist, and we got together and they look at the way, and then of course I said we put a little drop of ink. Then we also look at, when we take the core out, if the core has shifted from the edge. If the front is lining up at on the back end, then it is pretty sure they line up correctly. But if it gets shifted, when you cut, and then of course the outer needle come and close the tissue core, and then it has to cut it. You have to close the cannula and cut the piece. Even if a little bit of still attached, when you are pulling the needle, that that disturbs the core inside the notch. So this is a very challenging aspect that we had pathologists and the technicians work with us closely to ensure this millimeter correlation is as accurate as possible.
Thank you very much, sir.
Any other questions? This is a very challenging area. I mean, as an engineer, you have your computers, your equipment. Mostly you don't need much of approval from anybody. This one, you can sneeze without getting approval from the university and the patient. You have to consent the patients. It has to go through the ethics boards, and then the government approval. I mean, there are various levels of approval, and so you have to have you have to be very dedicated to do this, because one of the things when they hired me, was very, very specific. Late Dr. Gary Miller, he was the one who got me into this field. He said, listen, you are an engineer, you are coming here, and I don't want you to come and tell me I can cure cancer in rats. So you look at me, go, I know how to do that, Priya. So don't worry. We know how to cure cancer in rats. What we don't know is how to cure cancer in people, in men. That's why you are hired. So one of the things he told me was, when you do whatever you do it with the real thing, meaning don't go for cell lines and animal models and all those things. Do it with the human tissue, or do it with the human patients. That was the thing that he advised me, because we have enough people in medicine and sciences doing cell lines, animal models, mice, rats, pigs, whatever you want to call, dogs. We have. You don't need an engineer coming to do any of that work. So that was the advice. So thanks to that advice, all of my work were done with human subjects, the real thing. And so if you look at the publications I have done. So they were pretty confident taking it to the clinic and doing it because there's nothing else to be tested because it's already tested in human subjects. So that's the challenge. That's a very big challenge because, animal models, cell lines are, I mean, I'm not saying they're not challenging. They are challenging, but not as much as human subjects. You really, really, really need to work very hard to get even a single data point.
So yeah.
Anything else?
Yeah. So with that, I think we can wind up the Q&A session if you all don't have any more questions. Okay.
Yes.
So,
Yeah. Sorry.
yeah.
Little interruption. What were the difficulties you had, and how do you overcome during? I think you are very confident and very enthusiastic person in this field. So I think that will make some inspiration for us.
Yes. So the trick, I'll tell you the trick. I learned everything from scratch, medicine. So I wrote all of my protocols. I was not counting on the doctors or any other. I wrote all of my approval protocols from the FDA to local authorities, everything. So that is the key. So in order to do that, I had to study. I mean, I had to study from medicine from scratch. Because as I said, I mean, I didn't like the field. That's why I became an engineer. When I went to Ananda College, I used to walk by the general hospital, and I hate that smell, iodoform and all that stuff. I said, that's not for me. So, but I had to learn everything from scratch. So the writing is the hardest part in this field. If you want to take it to your university ethics board, you have to write the protocol. So there's a protocol, and then there's a protocol summary, and then there's a patient consent. The patient consent should be written at an 8th grader's level understanding. So, so the key to success in this field is you're not depending on a lot of other people to do this clerical work. So you left the doctors to do their work, and you do the bulk of the work. So that's the key. I mean, it's not an easy task. I mean, writing is not an easy task, especially someone like with my background. I mean, I'm an engineer, and so I had to learn the terminology and everything else from the very beginning, and that took quite a chunk of my time. But again, it's when you look back. This is not the only thing. I mean, I have done fair chunk of other work that is already been practiced in the clinic. When you look back, the reward is very good. I mean, it's there are patients who benefited from these technologies. So that's the reward. That's the reward.
Thank you very much. Lakshani, you can carry on.
Okay.
Yes. So once again, it's really inspirational having you on board, sir. So thank you very much, Dr. Werahera, for your valuable and insightful responses. And thank you to our participants for your engaging questions. So as we come to the end of today's session, it's my pleasure to invite Engineer Sapamal Varisundara, Operations Team Lead at IESL Electrical, Electronics and Telecommunicational Engineering Sectional Committee, EETESC, to deliver the vote of thanks. Over to you.
Good evening, everyone. Good evening, sir. And we have come to end of the insightful webinar today. Actually, it was rich in both technical and it was actually inspiring to all of us as engineers in different field. Actually, the consistency you have achieved and the things you have done actually inspired us. Very much thank you for your valuable time, Dr. Priya and Werahera, for sharing your valuable knowledge and the expertise on the ClariCore optical biopsy system and its role in real-time diagnosis of prostate cancer. So your presentation actually provided us with a fascinating perspective on how engineering innovations can significantly advance medical diagnosis. And we truly appreciate your time and your insights, sir. So apart from that, my special thanks also goes to IESL President, Dr. Kamal Laksiri, the past president of IESL, and his valuable coordination support for conducting you and doing this arrangement. So also, I sincerely thank to IESL current president, Engineer Kosala Kamaratani, sir. So his guidance on for doing these things. And also, my appreciation also goes to Chairman of Electrical, Electronics and Telecommunication Sectional Committee, Telecommunication Engineering Section Committee, Engineer Viraj Muthugala. Actually, he's the leader of us. So we are this year we have very rich presentations and webinars arranged for you all with a lot of industry leaders actually. This is one of that actually. This is third or fourth webinar. So also my sincere gratitude to the whole organizing committee of Electrical Engineering Sectional Committee and also the IESL Secretariat for giving us this platform to work on, and also for the tremendous support they are doing in parallel. So also my thanks goes to all the participants. Actually, there were many from abroad, and also many doctors are also there from Apex Hospital. I also shared them. So, and a lot of engineering enthusiasts was also there. So this, even though this medical engineering, biomedical engineering is a little new to Sri Lanka, so participation was very rich and they are enthusiasts. So, and with that, I invite all to be with IESL Electrical Engineering Section Committee for the upcoming webinars and participate in the ongoing discussions. Thank you very much, and thank you, sir, again, and we'll meet. Yeah.
Thank you very much. I have thanked all of my mentors and everybody here. And again, I want to thank you guys for taking time to listen to this. And thank you very much. And you have a wonderful evening.
Yes, so with that, we conclude today's webinar. Thank you once again to our speaker, Dr. Priya N. Werahera, and to everyone who joined us this evening. So we hope to see you again at future technical events organized by the Institution of Engineers Sri Lanka. And have a pleasant evening. Thank you and goodbye.